This method can help answer key questions in our understanding of genome regulation, such as how disease-associated sequence variants in non-coding regions affect gene expression and regulation by altering protein binding, and it can help identify those proteins.
The main advantage of this technique is that it does not require any sort of genetic manipulation of cells or tissues, no antibodies, and no prior knowledge of DNA binding proteins that might interact with the DNA at that locus.
Demonstrating this procedure will be Danu Perumalla, a technician from my laboratory.
After designing oligonucleotides according to the text protocol, plate 2 to 5 × 10 to the 5th lymphoblastoid cells or other cell lines in a T25 flask with 6 ml of RPMI 1640 medium supplemented with 1% pen strep, 2 millimolar L-glutamine, and 5% FBS. Incubate the cells at 37 degrees Celsius and 5% carbon dioxide. Use a hemocytometer or an automated cell counter to measure the cell density. Before the cells reach 1 × 10 to the 6 cells per milliliter, spin them down at 100 × G for 5 minutes and transfer them to a T75 flask with 25 ml of the RPMI 1640 medium prepared earlier in this video. Grow the cells until they reach a cell density of 1 × 10 to the 6 cells per milliliter, then transfer the cells to a T150 flask in 38 ml of the same medium and grow them for an additional 2 days. Next, spin down the cells at 100 × G for 5 minutes and resuspend them in 10 ml of RPMI 1640 medium. Then pour the suspension into an 850 square centimeter roller bottle containing 500 ml of medium and incubate the